Thanks. So we move on. We have kind of a subsession now, which is called vegetation and aquatic environments. So we will hear Yonghua and then Jonathan again. So we start with Yonghua.
Okay. Okay, good afternoon. I'm very happy to be here, and thanks Virginie, Ben, and Fabian and Neda for putting this together. I think it's really the first time I came to LSCE Institute. So I choose a style. My presentation is not really a research seminar as what I used to do for my own research. So a little bit overview of what I can see, maybe potential interactions with people here in this institute. So I'm Yonghua Li from CA Kadash from the BIAM Institute. So all my scientific career I've been working on lipids. So a lot of people don't know lipids or don't like lipids because when we think about lipids, we think about the cooking oil, the things we can put in the car. So often it's greasy and it's not really like pleasant. But so today I'm going to tell you, in addition to lipids play many roles in our cooking, in our cosmetics, in the paint, everywhere you look around in this room, there are things waxes around this chain, this rod. So they are all lipid products everywhere. But that's what I'm not going to talk about today. Okay. So what I want to highlight today is more about the role of lipids and biological function of lipids in cells, in living organisms, not only photosynthetic cells, but in all organisms. You cannot create a mutant without lipids. So just show you the importance, because there are a range of very diverse compounds that are present everywhere. When we talk about membranes, and essentially we are talking about lipids actually. The membrane lipids are phospholipid or galactolipids, but also they are form of energy and carbon storage. If we go to the field, the seeds, if the seeds have defect in lipid metabolism, the next generation won't be able to grow or germinate before the photosynthesis take over. So that highlights the essential role. But increasingly, we know that they also play a role in signaling and in connection to the environment, also for establishment of symbiotic relationships. And lipids also play a critical role. But of course, there's intracellular lipids. There's also extracellular lipids. But many organisms secrete lipids outside to the environment, either to protect them from UV radiation or from water, some waterborne pathogens. And they can also play a signaling role in communication between different population of insect or the fruit-based insect, for example. So just highlight the importance of lipids and why they are so important and what are lipids actually. Broadly speaking, they are anything that are soluble in organic solvent. But strictly speaking, when we work on lipids, we're talking about lipids that are derived from fatty acid and its derivatives. So in nature, there are over thousands of fatty acid structures that we can find that explains large biodiversity in function also, in physiological function or in industrial applications. But most free fatty acids are not free, so they are attached to a glycerol backbone and with different head groups. So these kind of chemical structures give them an additional property, like I show here. Just a single enzymatic reaction between the two structures can change something solid like butter to the olive oil liquid form. And if you think about this in real life, in our cells, and if you go to a -20, I don't know, in Northern Europe, and if you cannot do this adaptation of membranes, you could be really freezing, and that will not only disrupt your membranes, but also the membranes essentially supporting protein functions that can be photosynthetic proteins, that can be proteins that are important for biosynthesis. So this is the reason for that is because the different form of membrane lipids, they can form different biophysical forms, the kicks and turns in your membranes. We saw typically in the photosynthetic chloroplast that there's a thylakoid membranes form little turns. So these are different zones, there are subdomains that are enriched in certain lipids, but not the others. So how these are distributed, we know very little about. Just give you a few highlights on the physiological functions people start to show through mutant studies. So all the images here I show you, this is Arabidopsis plants, and these are in the ocean for phytoplankton species, and show that with any defect in a particular specific genes involved in lipid biosynthesis can make the plants less tolerant to salt, to heat, and to freezing tolerance. And there's a global, this is more climate research or in the real. These are laboratory conditions, these are in the field. So just to show you that for proper functioning of a given species, either plants or algae, they need to have this highly regulated system to allow biosynthesis and also lipid homeostasis in the cell. So in this general background, what do we do in our lab? So we have an overall question to study the use of photosynthetic cells, either in the single cell microorganism like microalgae or in higher plants. How cells, this is a drawing of one of the cells inside, how cells can assimilate CO2 through photosynthetic energy, and on interaction with nutrients. A few talks this morning, we already talked about carbon and nitrogen interaction. So one of the ways interacting also through cellular redox, because photosynthesis can produce energy, but the redistribution of energy and the redistribution of carbon inside the cell between subcellular compartments are very important for the final outcome of a biochemical composition of a given cell. So we particularly study photosynthesis and interaction with nutrients. Gilles will talk about tomorrow about photosynthesis and CO2. So today I'm going to mainly focus on once the carbon is converted to organic sugars, what can they make? So they either make membranes, that's very important. A small part, depending on environmental situations, can be diverted to form what we call storage lipids. That's a triacylglycerol. It's a glycerol backbone with 3 fatty acids. So we studied this in the general context of producing bioenergy or biofuel, but we also realized that, like I showed earlier on, the fitness of cells is very important. So if we want to use a given organism for any bioproduct production, we have to not only understand its own biosynthesis, but its incorporation to the cell division and growth. We also identify the major problem in scaling up, and I will show you later on, because the stability of a given phenotype. I think the first talk this morning, we talk about the metabolic or phenotypic plasticity. And so is it metabolism more flexible, or is it climation, or is it adaptation in the long term? So all these actually goes hand in hand. So I'm not going to talk about the secretion and the production of hydrocarbons in the cell, but algae as well. We discovered a new enzyme. David this morning mentioned briefly. So this blue light driven enzyme. So what's the significance in ecological setting? We don't yet know. In the oceans, why blue light flash on algae, why it produce alkene, very small amount? Is it a signaling molecule, or is it a structural component in the membranes? So that's still open questions. So about oil synthesis, I said oil is the same oil as we eat, as the fat in our body. So we, like many other labs, we know a healthy grown algal cell here is a green photosynthetic chlorophyll autofluorescence in red. And when you stress them, they make a lot of lipid storage in this lipid droplet, triacylglycerols. And we realize this situation is very dynamic. You can switch on this synthesis and switch off, go back to this original state very easily. So we use this as a model, as a proxy to study what enzymes and proteins that are involved for the either synthesis or degradation. And we did a forward genetic screen 10 years ago. We identified many factors, but the more particularly important, what we identified is even if you have the same genetic defect, it doesn't have the same phenotype. That's really dependent on the environmental factors, because my mutant can lose its phenotype if I have slightly different CO2 levels in the chamber, or if I, the night cycle have a different photo period. So that highlights the interaction. Finally, it's not, if we use forward genetic screening, we identify so many important proteins, regulators, but if we ignore the environmental signals that are helping it to form its final given result or outcome, it's useless because we could lose the phenotype. And this, of course, become even more important during scaling up if we put in the wild many conditions, light, temperature, and CO2, we cannot even control them. So how to understand the dynamic interaction between lipid metabolism, lipid biosynthesis, and environmental signals that the cells are exposed to? It's one of the key questions. It's also the basis of our current PLPR project, just started in our lab. So I said already, underlying, so if you think about environmental signals and the genetic makeups, how we change that? Actually, similar, we found actually from many study of, I don't go to details, but study of many of these components involved in either regulators, kinases, or in the redox shuffling between different subcellular compartments. We highlight that redox is a general signal that can communicate and connect environmental changes to the nucleus to switch off the expression of the genes. So currently we are doing a range of genetics, also different transcriptomics and proteomics, physiology with changing environment monitored. We don't do much field work. We really do small scale, lab scale work. And with new tools in genome editing to be able to really dissect what's the interaction, how we can control that phenotype. So we can use it for scale up. I think this is more true, as I said, in a ever-changing environment, because unpredictable. If now it's 2000 microeinstein outside, or it's going to be 100 microeinstein outside the light intensity and the light spectrum also, we recently showed that the red light or the blue light can also impact the carbon partitioning and carbon storage in algal cells. So these are all current research in this general setting. And we also start to have different collaborations with people in BIAM or outside the BIAM Institute on the importance of lipid and the membrane adaptation for supporting or maybe improving radiation resistance or for climation to more heat waves, and also for different kind of radiation and the light and temperature changes, and with the ongoing work. So I hope I have convinced you that lipids play a central role in connecting not only climate research, but also in production of bioproducts and bioenergy. And I'm sure these two different areas are very goes hand in hand. It sometimes might not be so evident from outside, but through the study of triglycerolipid homeostasis, we realize it's true. We cannot be dealt with separately, so we have to work together. And this is, I'd like to thank all peoples of our team. And this was an old picture. We should have a maybe better camera this year. It's a bit fuzzy, but these are all the students and CDDs who have worked with me in the past 15 years on lipids specifically. Otherwise, these are current members of our team. So I thank you for your attention.
So we switch to Jonathan about allocation, and then we ask questions.
Now that we can continue as we were before. So we go forward to okay, that was one. So just to continue from where we were before, that we have these three rings that are much more than width, but there are also isotope, and there is also this possibility to look at allocation. So here's where we enter to the bottom of this circle here. And so in addition to the rings in the roots, we could also measure the growth of the core root at the same time than the stem and branches, for instance. So we have been installing this setup, dendrometers on roots and stems, and also coring the roots and stem of trees, where we can go back in ancient trees, we can go back to pre-industrial times. And then we have the relative growth investment in coarse root and in stem. And we are expecting that we could make the link between coarse root growth and fine root mass. That's the link that we want to make. And with that, we basically want to get a whole tree perspective. This is like a macro tree still. We don't go to the molecular level, but we want to get a whole tree perspective below ground plus above ground. Next. And here is an example of a measurement of a setup that we have in so in a given site we put a dendrometer in the stem and roots. Here there are 3 trees, so black, red, and yellow, and we see that the stems of the 3 trees, which are the thick lines, basically in July, at the end of July of this year, when we installed the stuff there, this is in Fontainebleau Forest in Fagus, we see that the stems were basically are flat. They have almost no poles, whilst the roots are growing and also show the transpiration daily dynamics. So, and then at the same time, we measure soil moisture, like in the bottom, the gray shade, that's the soil moisture at 10 centimeters, and the black line is soil moisture at 50 centimeters depth. And then also at the same time, we measure the atmospheric humidity, and in a way that is the vapor pressure deficit. So with those method forcing, we can really interpret the growth fluctuations, and also that are due to basically the size fluctuations are due to growth, and also the reversible fluctuations that are due to water transporting the tree. But here the interesting thing is that unexpectedly the tree is really midsummer transpiring a lot, but it's not growing in the stem. It's investing in the roots. So next, here the idea is that with this setup that we have the pair stem and roots, we can do apply the allometric equation, and we can track the growth of the stem in relation to, or growth of the root in relation to the stem. So the idea is that we can get this parameter, allometric parameter alpha, in the allometric equation, and probably characterize for each tree, and then on average for each site, how trees are allocating to grow roots, and also to stem and root, and also what are the deviations at the seasonal level and interannually. So we are starting now with the setup, and this is a bit the idea of how we want to summarize this alpha parameter, the allometric parameter, in different biomes, different species, and different plant functional types. But also here, when we go to wood growth, which is the other important process for carbon cycle. So first, we concentrate on photosynthesis a lot in the leaf, but the process of wood formation is not really modeled yet in these large-scale models, and we are measuring this in order to model that in the project. And so here, the idea is that there are also wood functional types, because there are different species. There are species that are like the conifers, they have tracheids, and then there are diffuse pore wood, and also ring porous wood, and that have different connections to different phenologies, and also different functionality with water. And the dendrometers do really show the different dynamics as well. Next. And so we are measuring this in sites. In here we have a global picture of all tree ring sites in pink, and then we have the flaxnet towers in blue, and we see that basically they are all over the place, but they are not necessarily collocated, and are 2 different communities. And what we are doing in CATES is putting the 2 communities together, and we have selected some super sites, what we call, and which are the yellow dots. where we are measuring trimmings, all the set of trimming, the isotope, the wood row allocation, and xylogenesis wood formation in the same place where there is a flat tower. So we measure this in the footprint. And then we also have the dendrometer set up with, as I showed you before, in different places to cover more area, and that is the yellow crosses. So we see that we cover tropical, subtropical, and temperate forest in the southern hemisphere, and we also want to do the same in the northern hemisphere. So we're slowly advancing, and in different transects through Scandinavia to South Africa, passing by the tropics in the Americas, and we also hope to do something in Asia with the same transect, north to south. And the idea there is to compare also dynamics in the northern and southern hemisphere. Next. And here there is an example of different setup that we have now in tropical, subtropical, and then temperate rain in the northern hemisphere and southern hemisphere. So this is how the forests look. And the idea is that with this we will get a bit of an idea of how allocation, at least the coarse root and stem, work over these biomes. Next. And I think that is in short what I wanted to show about the allocation part. And here the idea is that also we are doing in Illinois in the US. We have this setup where we have data, very detailed data on fine root dynamics. So we will connect the course with the fine root dynamics in this site in the Arboretum, Morton Arboretum in the US. And that we expect that we can see if we find this relationship could be between coarse root and fine root, which is this healing half somehow of the MPP that goes to every year, and that we know very little, and let alone that their long-term response to growth change factors. So we expect that with this setup, we can pull some information on the long-term changes in allocation of wild trees, that can be in a way become a benchmark for what we simulate in orchid. Thank you.
Thank you, Jonathan. Do you have questions for Yonghua and/or you can ask questions online too. With a button with a raised hand. No, so there is time like quarter to 20 minutes for a general discussion on vegetation. So we have seen lots of common interests, so we can kind of brainstorm whatever you want. We can another break and go to the restroom. I think everyone is a bit tired. I don't know. Projects are ongoing, and we hope the instead will be funded. And so for the stomatal conductance, I was very interested in the presentation by Natalie. It's clear we are interested, and we are looking at new models of optimal stomatal conductance. So I think there is room for improvement. Nicolas?
Natalie is online. Okay. Well, we were wondering about the connection and envelope scale within global climate model or such as or global land surface model such as 40. Do you have some work already under partnership with colleagues that work at that scale? You mentioned that activity, but we were wondering which group you may be involved. Sure.
Natalie, did you hear the question?
Not really. Sorry, could you repeat a little bit?
Oh, actually, you mentioned some, I don't know if it was ongoing or planned collaboration towards global land surface modeling regarding the stomatal conductance. And so we were wondering which team where you were involved.
No, I'm not involved at the level of the canopy or even of the field. We are doing only in a chamber right now.
Yeah.
But I will be very interested to see, for example, if we have an increase, or let's say if we can adjust the parameter of the conductance with model plants or other plants, because we are working with several type of plants, how we can improve, for example, the parameters that are put in the model. I have no idea. I'm not expert at all in the model.
No, it was, well, it's good to know. I think as mentioned Fabian, I think.
But the good thing is for me, is that what will be important is like how we can improve the model, or how we can modulate this parameter in response to a specific factor, because we know and we can have some, let's say, specific ecotype that does not respond, and just to validate what we are having in the model, I will say.
Laurent, so you have to speak louder so that the people online can hear your questions or comments.
When you take, when you go from,
You can speak normally, the mic is on the.
okay. So when you take into account nitrogen use efficiency, for example, you can have a broad variation between plants. So how can you implement this into model? What is the range of variation, for example, for wheat? Did you identify with the different wheat you identified, and how you can, it's a very precise parameters. So if you want to implement into your model, how can you take into account the diversity of plants, of crops, or so to use it finally?
Regarding the parameter you mentioned, so far we account for this range of values for the functional type we can have. Not typically for wheat so far, but more like also. And we between PFT across PFT, it's a change of parameters. For sure, if you then focus on wheat varieties, it's not at the scale we are working on, but then we may have so it's more encoded that it's more involved in the traits.
Yes, yes,
we have
because
a
it's
sub-PFT diversity.
indeed one of the limits that we have now in the model with the PFT representation is that for each PFT, we have some parameters that are fixed. So in fact, what we do, we suppose that we have the same plant everywhere, so as a clone of the same plant. And so this is why we and all the community of the GM, there is now effort to try to go from this representation of by PFT to a presentation which is more trait-based. It's something which is more continuous orientation than we have in the PFT. For example, in this case, so there is different kind of approach. There is some approach which are based on try to find some empirical relationship between traits and for instance, the environmental conditions. Another approach is to try to find some ecological optimality principle. So, for instance, if we talk about nitrogen use efficiency, we can say that the plant will try to optimize their nitrogen use efficiency, and so it's what we try to do, and it's part of the project in green scale. Something that we begin to try to implement in the model is the coordination of photosynthesis. So which in fact try to find this optimum nitrogen use efficiency. So it's another way to try to improve this representation to have something which is more continuous. So, so it's something on which we go toward. Of course, at global scale or at large scale, obviously we will never have the observation at sufficient fine scale. So it means that in this case, we need to do some hypotheses about the fact that so try to find such kind of relationship between the trade and the environment, or try to find this optimal principle to be able to have the trade that vary in a more continuous way, to be indeed be able to take into account the fact that, for instance, you will have, well, for crop, of course, it's more complex because it's human driven. It's not so. So in this case, of course, it's more complicated. So we mostly look to natural vegetation for this case. Of course, if we have crops, we need to take into account the fact that it's not natural at all and that we cannot use such concept.
Okay.
It's more a general question to young people. So you've seen how good we are modeling the dynamics of plants at our global scale model. So we are compartment based. Jonathan mentioned one of our key issue is the allocation of carbon once you have done photosynthesis, and between above ground, below ground. So what do you see and anticipate that could make us the next nice step in terms of complexity between having 8 carbon pools like above ground, some hardwood or hardwood and bouquets, sapwood and hardwood, leaves, trunk, branches, and then the roots. Should we include compartments which are the lipids, and should we have something new? What do you anticipate? There's one question we don't model properly, at least for us. One thing which is the legacy effect of, let's say, a drought given. If there is a drought 1 year, the plants probably don't fill enough the reserve and don't, it has an impact the next year. We don't simulate that very well, I mean, at least not us, and it's a big challenge in our community to have this. So do you see major change, whether it's on the nutrient, so there was talks on the nutrient, phosphorus, there is an ongoing collaboration on the phosphorus that Daniel has mentioned, but do you see other paradigm to change in the way we model? And you may say, oh, don't think, oh, it will be too complicated computing time, computer-wise, because we are entering a new world of artificial intelligence with emulators. So let's imagine we can do everything. And so what would be your suggestions of new concepts to improve? Or do you have, I mean, I'm just asking.
Well, but we can ask the same question to you. What do you need? What kind of parameters, what kind of processes do you need to know in detail? What is the and at which scale do you need to know to take into account the biological part in the models?
In this model, we will never be, I don't think, at the cell scale, but we are more, I would say, we can go maybe to the leaf level, but it's really the minimum is the leaf level, and then we go to the plant and then to the landscape, where it's our main application for the landscape. But the plants, and even we start to have leaf levels, we have shaded leaves, light leaves. So for us, as I said, this legacy effect, this changing functioning and these acclimatations, whether it's phenotypic, whether it's for plants that have a large turnover, whether it's genetic adaptations. Arid ecosystems, we are very, very poor into the modeling of the response of grasses in arid ecosystems to. So Yeah, you can see it from our side, we are more has empiric because we don't know enough what you're doing. And if you from your side you see, oh, you should do that. Yeah.
So first Nathalie and then Laurent. Nathalie?
I have a comment. I just wondering, because you say that you modelize that most of the climate or the CO2, but when you are doing that, and if you are taking account the history, so let's go back, and if you use this model, does it fit or not? Because we know that there was a big change in adaptation and plasticity for the vegetation, for example. And does it work, or how you take account the modification of the vegetation in your model?
What do you mean adaptation over which time scale?
Yeah.
Geological?
Let's say on the let's say if you take the model like let's say 100 million ago, does it fit or not? How you take account that, and can you use that to project what will be the future? This is
Yes, so in fact, what we did is the opposite. That we had a PhD, so Julia Bress, that because indeed in the lab we have a group that work on paleoclimate, and so we try to work with them to see the impact of vegetation on climate, etc. And on this PhD, we work on exactly on what you presented on the chain of angiosperm, and the fact that we had an increase of the number of stomata, and so what we did is that we had some measurements from the fossil data. We try to use this data to parameterize the change in the stomatal conductance in orchidea at different time. And so we define some paleo PFT, I can say, and she used it first to see the impact. So we had a past angiosperm that was a little stomatal conductance and lower photosynthesis, and she looked to the impact on the climate itself to see how it the climate on the past, but also what she did, and that was something very interesting. She because we can use orchidea in dynamic mode, which means that we can let the model determine which vegetation would be possible to live with some kind of climate. And so she put in competition, the modern angiosperm with this old angiosperm. And what she was, and the conclusion is that so the period it was in, you remember, 140 million years ago. And that the old angiosperm was competitive with the new one, but now if we try to use it for the present, we can see that they are not competitive at all, and in particular, for instance, we could see that the forests, the tropical forests, for instance, could not grow. It was a very interesting result. So it's something indeed which we prescribe something that we have in the past, but as you said, something which is interesting, and I was talking about this question of optimality of process, and for instance, coordination of photosynthesis. And then it's indeed interesting because we can try to see using this coordination or optimality, try to reconstruct in particular with the past CO2, which is the most important value factor, to see if we are able to simulate the decrease of stomatal conductance. That is something observed. So it's something we didn't do for the moment, but we would like to do in the future. So indeed, we can use the past climate, and if we have some information about past vegetation, to try to see if we use this well, plasticity of trait. Can we are able to reproduce something that was in very different climate or simple conditions?
Okay, Laurent wanted to add something?
Just to know, in your model, you take into account the fact that the climate seems to be more and more hierarchic. So there is some amount of water, for example, in some area, but which will come in a shortest period of time. So can you predict this? Can you predict the impact? And what kind of vegetation will be adapted and the move of vegetation?
Yes, so indeed, because in the model we have the footprint in the soil, etc. So we can indeed take into account the fact that the water arrived, a lot of water arrived, and a lot of water, in fact, will be drained, which is not the same that, and because we have a relatively short time step in the model, the time step is half an hour. So indeed, we are able to simulate the effect of the extreme events on
We do that, but it doesn't mean we do it well.
Yeah, it's another question.
At least
You're right.
we do it. Maybe other question I like full of bunch of questions like that. So for us, what we do, we conserve. We are conservative people. We conserve energy. We want to conserve mass, so we conserve carbon. But for the leaf, we have some photosynthesis. We don't know if we do thick leaves or very thin leaves. And so we have a parameters, SLA, but how these parameters may evolve through climate warming, under drought conditions, these kind of questions, how the leaves really or the plant do they do thick leaves or invest more in the roots or not, depending on the weather condition. It's things that we need to model properly, but where maybe some experiments can be at the leaf scale or in the laboratory can be helpful for us. And as for me also a general point of view, you have to think we have a model, it has different scale of applications. So if you have questions like you have a new process, is it important at the larger scale, like can it impact that? Then you can always send that question to us and we can see whether we can test it. But in principle, we have a model to test hypotheses at larger scale. I'm not saying we test it well, but at least that's to test at larger scale what is the impact of having a new pool of lipids in the plants and making, I don't know, people question.
We have one question from Jean or one comment?
Yes, so I think that's a really good question. Well, I'm not sure we know how the vegetation is going to react to increasing CO2 concentration. And I believe there are competing projects in this fair carbon call that are going to address this point.
There will be competing projects in fair carbon.
It might be, yes. I had a question about the plant functional types. How do you see the future developments in orchidea? Would you like to implement more plant functional types? Is there a way to decrease their numbers? How do you deal with forests like the Mediterranean forest, which is composed of like pine trees, which are evergreen, and oak trees, which are deciduous? They correspond to two different plant functional types. How do you deal with that? And if you had a suggestion on which plant functional type would you like the biologists to work on as a model? How would you if you had to define a model organism to study for a biologist, which would be helpful to parameterize the plant functional types, what would you suggest?
Maybe I can repeat what we do separate evergreen versus deciduous needle leaf. We have two different plant functional types. For Mediterranean forests, you correct me, Nicolas or Fabienne, I think we do not represent well the transpiration and the water transfer links to, especially during the summer, a drop with the drought. We do not represent that well for the oak trees of some of the Mediterranean forests where we have flux data. So this is something we would like to develop. And for these water stress and how the plant responds to water stress in these regions, and then whether we will go for more plant functional types or reduce plant functional types. We want to be more realistic, so we want to find some equations. So there are approaches like Colin Prentice, which tries to develop unique parameters that are, instead of having parameters as a function of plant functional types, these parameters become a function of climate or soil or condition. So that's one way to go. Or the other way is to go with as many species as we can describe and have maps of parameters, which are specific to species. I'm saying that there are two competing developments in the community, and
I think that's too much to ask to the biologist. We can't study all plants at the same time for all parameters. But for example, the drought response, do you find similar acclimation mechanisms in different plants? Do you parameterize differently, for example, the stomata control in a grass and in a tree, for example?
the equations behind are similar, but the parameters can be and they are already optimized independently.
And you can have some parameterization that are PFT dependent.
Yes, the
But
phenology is not the same for trees, for grasses. And now we even start for grasses to have densities of grasses. So we do want to model the density of grasses to represent their behaviors, like we have density of forest. So yes, but maybe for us, we need to focus on one, and Mediterranean ecosystem is a good one.
Okay, it's good. I see we have a lot of things to discuss. Okay, so I see Ben and Jonathan, you still have, but we have, there is a nature-based solution talk by Nicolas, and then we can talk again. There is another discussion session just after Nicolas, so stay tuned and we will have questions by Ben and Jonathan.